The contributions that this research is going to provide will pay dividends in terms of moving our DNase I program forward into the clinic and in the design and execution of the clinical trials that are coming down the road.
Welcome back, and thank you for joining us today for another Virtual Investor What This Means segment. My name is Janine Thomas. I am CEO of JTCIR, and I will be the moderator. Today we are featuring Xenetic Biosciences, and I am very pleased to be joined by Dr. Reid Bissonnette. He heads Translational Research and Development at Xenetic. Welcome, Reid.
Hello.
Before we get started, I just want to remind our audience that Xenetic Biosciences is publicly listed on NASDAQ and trades under the ticker XBIO. And during today's discussion, the company will be making forward-looking statements, and I encourage everyone to view the company's latest SEC filings on their website at xeneticbio.com for the latest information. So, Reid, we're going to dive right in. So, to get started, can you please provide an overview of Xenetic Bio and your innovative DNase I platform?
Sure. So Xenetic Bio is a small biopharma company based in Boston that is progressing through its oncology drug development pipeline, the nuclease DNase I. So DNase I is an enzyme. It breaks down DNA in a variety of ways. Physiologically, DNase I is responsible for cleaning up cell-free DNA, preventing that DNA from becoming a health issue. For example, cell-free DNA is implicated in certain types of autoimmunity like lupus. So why DNase I? Xenetic Bio is developing DNase I. It's also called XBio15 as a drug targeting a physiological structure called neutrophil extracellular traps, or NETs. NETs are expelled by activated neutrophils, a type of white blood cells. For context, neutrophils are the body's tip of the spear for dealing with infections and other insults. NETs are expelled by activated neutrophils through a process called netosis. Now, these nets are web-like structures, as you might expect from the term nets, composed of extracellular chromatin, DNA, that are decorated with histones, enzymes, and other proteins derived from the neutrophils themselves. Now, nets are beneficial in the case of things like microbial infections, where they trap the microbes and hold them for destruction by the immune system. In cancer, however, tumor-associated nets can create a host of problems. Nets are mostly pro-tumor, meaning that they promote tumor progression. They can promote and facilitate metastasis and can contribute to an immunosuppressive tumor microenvironment, which really hamstrings the anti-tumor immune response and the ability of the immune system to control and kill tumor cells. They can physically interfere with the ability of immune cells, such as killer T cells, to detect and engage these tumor cells. Nets are known to contribute to resistance to chemotherapy, immune checkpoint inhibitors and other immunotherapies, and radiotherapy. That's why Xenetic is pursuing DNase I as a means of targeting NETs to improve patient responses to cancer therapies, and in particular, chemo and immunotherapies. To that end, we are accumulating preclinical proof of concept and mechanistic data that demonstrates that DNase I, by targeting and destroying NETs, can significantly improve the efficacy of several cancer therapies.
Appreciate that background, Reid. So Xenetic recently announced that it had extended its research agreement with the University of Virginia for the advancement of its DNase-based oncology platform. Can you provide an overview of the agreement for our audience?
Sure. What we have is what's called a sponsored research agreement with Dr. Alan Sung and his lab at the University of Virginia, or UVA. This agreement, which I see more as a collaboration than anything else, was initiated in December 23. And the first part of that agreement, or tranche 1 as we call it, was completed around June 2024. In that first tranche, Dr. Sung's lab conducted studies looking at the ability of DNase I to improve the response to standard of care, or SOC, chemotherapy in preclinical models of primary and metastatic colorectal cancer, or CRC. Dr. Sung and his postdoctoral fellow Hongji Zhang already had experience in models of CRC, but the one we chose to use was a model called CT26 that is representative of the most common type of CRC, which accounts for about 85% of CRC patients, and which is referred to as microsatellite stable, mismatch repair proficient, or MSSS/MMRP. We did this because this population either doesn't respond to or responds really poorly to the current crop of immunotherapies. And after that last line of chemotherapy, there really isn't much else. But because NETs are implicated in the resistance or non-responsiveness to chemo, We believe that DNase-1 could change that by eliminating nets. So, long story short, that effort was very successful, and now we have a really, really good data supporting the clinical investigation of XBio15 DNase-1 in combination with standard of care chemotherapy in this subset of CRC. And so the indications we're thinking about include CRC as well as pancreatic ductal adenocarcinoma, or PDAC. The next phase, or tranche 2, will focus on investigating combinations of DNase-1 with what are now commonly used immunotherapies called immune checkpoint inhibitors, or ICIs. We'll be looking at ICIs targeting the immune checkpoint CTLA-4, for example. Examples of that you may have heard of are the FDA-approved CTLA-4 inhibitor, the antibody drug ipilimumab, or Yervoy. We'll also be looking at ICIs targeting the immune checkpoint PD-1. Some of the examples of FDA-approved PD-1 inhibitors are the antibody drugs nivolumab, or Opdivo, and pembrolizumab, or Keytruda. Based on our preliminary data, we're confident that we'll get some compelling data in support of going to the clinic with XBio15 and ICIs, immune checkpoint inhibitors. And we will be performing some very interesting mechanism of action studies to determine what the elimination of nets by DNase-1 is doing beyond just chewing up nets. We will also be developing biomarkers that allow us to select the right patients and pharmacodynamic markers to track response and gain more mechanistic insight.
Well, Reid, certainly very compelling. And you know we are here for a virtual investor, what this means segment. So I have to ask, what does this agreement mean for your overall development strategy, your pipeline, as well as Xenetic?
So this agreement and the data that it's generating is a key component of Xenetic's development strategy. First, I want to say that it's a privilege working with Dr. Sung and his team. Dr. Sung is a leading researcher in this area of nets and netosis, and he has interests in CRC and other gastrointestinal cancers like PDAC. He's a surgical oncologist, so both a surgeon and a clinician scientist, my favorite kind of MD, and he's the chair of the Department of Surgery at UVA Medical School. In addition to that, he has developed a model of metastatic CRC, and this is the key thing that is crucial to our understanding of how DNase-1 can benefit patients, and he has a great deal of experience in this field. So the contributions that this research is going to provide will pay dividends in terms of moving our DNase-1 program forward into the clinic and in the design and execution of the clinical trials that are coming down the road.
Appreciate that, Reid. Congratulations on all your progress. Exciting times for Xenetic and your DNase platform. With that, this concludes our Virtual Investor What This Means segment featuring Xenetic Biosciences. I'd like to thank Dr. Reid Bissonnette for joining us today, and I'd also like to thank our audience for your time and attention. For more on Xenetic, visit their website at xeneticbio.com and sign up for their email alerts to stay up to date on the latest developments. You can also follow the company on X, LinkedIn, and Facebook. And for all things Virtual Investor, visit virtualinvestorcode.com. I'd like to wish everyone a great rest of your day.